So then this is my team right now. So we are mostly a computational team. We like to call what we do computational microscopy, or how computers can be used as a microscope to look into biological objects. And this is the tool that we develop, that we work with, is called a molecular dynamics simulation tool, and we'll talk about that in a bit. So essentially, the evolution of these kind of simulation started. Let's start it simple. Keep it at 1990s. Of course, started around 1977, late 1970s. But then biomolecules were not that interesting. So gradually, we started around 1990s with very, very small systems like lysozyme. Major milestones were achieved all along the way. Nowadays, so much so nowadays, we can simulate an entire cell organelle in all atom detail, which will possibly be the last slide of the talk today. Normally, I start with that if it's a science talk, but because it's a methods talk, we'll possibly end with that. Primarily, the tool that we use is called a molecular dynamics simulation. So this is the tool that was developed by Klaus Schulten, my advisor. So in molecular dynamics, what we do is, the concept was not discovered by him. The concept was discovered way before. What he came up with was one of the very popular software that is used in the community right now. So you take all of the molecular dynamics is actually a very, very simple thing. You take all the chemistry of a system, which we know that electrons are delocalized between nuclei. And the first major approximation you make that they're not. A molecule is made of balls and strings. Each atom being a ball, and each spring is a bond, and the strength of the spring is tells you how strong the bond is. It's weak or strong. Then you essentially calibrate that into an energy function that we call force field parameters. And now you take a molecule and you start evolving this molecule following normal Newton's equations. And then what the simulation does is, it ends up optimizing the energy of the system till it reaches some sort of an energy minima. So at one level, you can think of it like a way of sampling this energy surface, visiting the energy surface that you're creating by simplifying the chemistry of the system. Of course, there's a subtlety here, which is it's not, even Newton's equations at the molecular level are not very straightforward. There is a stochastic term to it. There's noise to it. Normally, if I push something from here to here, it's a classical Newton's equation where there is no noise associated. It's very deterministic. If I want something to go from point A to point B, I can make it move from point A to point B without any sort of noise. In the continuum world, in the macroscopic world. Unlike that, in the microscopic world, there's a lot of noise that a system has to mitigate, which is the reason of heterogeneity often when we think of dynamics. There are multiple ways in which I can go from A to B, and that noise, that uncertainty is also a part of Newton's equation, using some sort of theorems called fluctuation dissipation theorem that we often don't acknowledge as much, but it is there. In statistical mechanics and molecular simulations, it's always there, and that continues. So now the exciting news for this area is right now, the first Exascale computer, we are in a DOE lab, has been set up in a DOE lab, namely in Oak Ridge, which allows us to do 10 to the power 18 double precision calculations per second. So the equation that I write here is an equation for, this equation has to be solved for all the atoms in your system. So you can understand, larger the number of atoms, more expensive this simulation becomes. So now we can in principle do 10 to the power 18 calculations per second. So this is where I ask my first question to make sure that my audience is staying excited. What does the number 10 to the power 18 mean to anyone? What comes to your mind, 10 to the power 18? It's a very weird number.
It's 5 orders less than the Avogadro number.
Exactly. So that is what I think. So I found someone with. I think I would have gotten that answer. Anything else? Okay, 5 orders of magnitude lesser than the Avogadro's number. Any other, what would that mean? That's a very good. I think of it that way also. That means it's kind of a micromolar solution. In a way, like one can think of 1 mole has 10 to the power 26. So that means in principle, one can start simulating systems that are as concentrated as micromolar or so, that many number of atoms. Now then, this is the bigger problem in the area. So if you think of computing time, like the time that is taken for CPUs to run, very recently a paper came out that says that it is proportional to n being the number of atoms in your system. Forget this part for the time being. T real is the total time of an observation like of a real biological phenomena or a chemical phenomena, however long that phenomena takes. You have kinetic measurements. Delta is your smallest time step for solving this molecular dynamics equation, because as I said, like it's a Newton's equation. You need to discretize that, and then you need to take time steps. So the smallest time step for Newton's equations, turns out we take femtosecond scale time steps, because that is the essentially the fastest possible spring is that of an oscillation of a carbon hydrogen bond, which is of the order of femtoseconds. And delta is essentially the number of flops of your V is the number of flops of your fastest computer available today, which is 10 to the power 17 flops. Now that has become 10 to the power 19 with exascale computers. So if you plug all of these things in, let's say you want to simulate an entire cell that is a billion atom, roughly a billion atom, and you want to simulate the process of a cell doubling itself. The first step, like living things, what is the first reason of life? What is the first activity of life? One is to sustain, and the next one is to grow. That's the basic. These are the two things a simple cell is going to have. You'd like to live, and then you like to grow or divide. So essentially, living means growth. So a minimal cell that, for example, the Craig Venter Institute has created only does two things: live and grow. They actually grow and divide. So essentially 2 hours is for a simple cell is a good time for seeing a cell divide. So now then, if you plug all these numbers here, it will take you 10 to the power 9 years to solve this problem with molecular dynamics simulations, with traditional molecular dynamics simulations, with the best of computers today. Of course, it's untenable, but I just wanted to, someone for the first time has created this very dramatic shortcoming of doing everything from first principle molecular dynamics. Why would you do that? Okay, might be then you can make arguments. You can say, okay, I am not very interested in a whole cell. A protein is a lot smaller than that. A protein in a membrane and water would actually come down to, let's say, the biggest will be 1,000,000, 10 to the power 6. So you lose 3 orders of magnitude here. That still brings you down to 10 to the power 6 years. You can say, I'm not very interested in 2 hours of doubling time. I would rather be interested in an event that takes milliseconds to seconds. So you essentially from 2 hours, you lose again 3 orders of magnitude, right? So because one hour has 3,600 seconds. So 2 orders of magnitude. So that brings you down to 10 to the power, how much? 10 to the power 4. Okay, and now you have 10 to the power 19. Still, that brings you down to 100 years. And then you can say, okay, I can come up with algorithms that are an order of magnitude faster than this. That still gives you 10 years. The best case scenario is 1 year. But 1 year on a national supercomputer only to yourself is untenable.
Question.
Yeah.
So how important is the time step being equal to the example of femtoseconds you gave? What resolution do you lose? What accuracy or precision if you change the time step?
Correct. And odds are
have it flexible. I mean, maybe there's times during the simulation. I don't know how stiff the systems are, but
so you're correct. So like if people choose to do that, right? Often people choose to lose this to make it an order of magnitude faster. What you lose is some sort of reorganization of hydrogen bonds, for example. That happens over time scales of picoseconds, so to speak. Those, if you choose to lose, let's say the hydrogen bond reorganization, then it's fine. Like you can say that my model has somehow I won't be already. I've made a huge assumption in my first slide, right, where I say that electronic degrees of freedom are all integrated into a ball and a spring model. So most of the quantum chemistry is gone there. So we can say, okay, fine. Let's also remove some of these fast changes in the system. Let's not incorporate that. And that gives us a little bit of in certain cases, you don't want to do it, but a lot of people do that. Let's say you want to simulate the entire genetic machinery, which is like really, really long, lots of atoms. You cannot do it with this. Okay. So, or a viral genome, so to speak, it's smaller than that. Okay. So now that's where the innovation also comes in from our team. So we normally don't lose resolution. What we do is we try to merge experimental data with molecular dynamic simulations and in a way accelerate movement by imposing data from experiments. Hopefully everything was moving there. Just wanted to show you. So what we say is that the underlying assumptions in these kinds of models is nature in principle doesn't know anything. So when it does a millisecond scale or a second scale dynamics, it has all the time and a much larger search space to select from. On a on a computer, we don't have, as I showed you, it's very, very difficult to recreate the search that nature performs to fold a protein, to bind, unbind to something. However, we have information that comes from the amazing structural biology experiments that we get to see here. And that only is growing right now, be it mass spec. Here we are telling NMR, crystallography, cryo, FRET. Why not use this information? We know that these structures are biologically relevant. We assume that these structures are biologically relevant, that these have a functional significance. So therefore, these structures should definitely be a part of the dynamics that my molecular dynamics simulation is actually looking for. So why not impose that information? So rather than nature had 1 full minute to reach those structures, my 1 full minute of real time, which in my last slide would have still taken me 10 to the power 1 or 2 years to simulate, to at least see one event. Rather, why not impose that information? And so that's what kind of accelerates things for us. And we'll gradually, so normally I leave it here and say that let's go on to do science about it. But might be today, if it is a more technically interested audience, which I'm assuming it is because it's a national lab and we're very interested in methodologies, I made it a methods talk and we'll take you deeper into how we do this. Otherwise, I'll take you back to my 10-year talk that I gave last week and show you scientific applications of that. So, is it okay to be more methodologically aligned today? Is that something that we would be okay with? For everything, like, yeah, that's why. Because again, I'm trying to gauge the audience here. But it's okay. Let's try to make it fun. If you don't like it, let me know right away and we'll switch. So this is the point of decision making. So okay, now then let's dive into how do we do this thing. Okay, so the first thing is we came up. This was something that Klaus Schulten, Hakim Frank, and Elizabeth Villa was then a graduate student when she worked on this. Now, of course, she's in UCSD and setting up her, learned electron tomography in Germany from Wolfgang Baumeister, brought it to this country. And all of us are actually now doing electron tomography. But three of the original people around 2008 decided to kind of merge experimental data with molecular dynamic simulations. In fact, the history is a little bit older than that, where someone called Charlie Brooks had also done things about molecular dynamics, how to use experimental data to accelerate molecular dynamics. So what I'm showing you here is a low-resolution cryo-EM density, really 2008 class cryo-EM density, so to speak, of a protein in one conformation and the crystal structure of the protein in another conformation. In principle, for this protein to go from one conformation to another, it would have taken milliseconds, if we did not know that there is this other structure that exists or this other low resolution information that exists. So what we do is we take this low resolution information, convert this into a Coulomb-like potential by using this formula. Does not matter what this formula means, but at least see that this additional potential is linearly related to the three-dimensional form of this electrostatic potential. So of this cryo-EM potential. So we take the cryo-EM density, break it into grids. Using those grids, we compute something called phi. So this is basically a discretized formation, a discretized form of the density projected on XYZ finite difference grids, very simple grids. And then we determine forces that those are the derivatives of this potential with respect to position. So in other words, an atom is not just feeling a force from chemistry, from the force fields that we showed introduced in the previous slide. It also receives force that are derivatives of essentially the gradients of the information. So therefore, the structure gets pulled into it. That accelerates this by orders of magnitude. Those 10 to the power 1, 10 to the power 2 that we're talking about is gone now. So now this was what is called molecular dynamics flexible fitting kind of simulations. Next thing is, these were very, very good back in the day when the density was quite low resolution. And what I'm showing you in the bottom is a slice of the density. Essentially, you see it's very, very, so this deep blue regions are local minima of the density features. And they were quite smooth, quite degenerate in older densities. But newer densities start like circa 2013 or after are much higher resolution. And the potential that is generated out of these densities have a number of minima, have a number of features that look extremely degenerate. And molecular dynamic simulations, or anytime when you move on the surface of a potential, you run into this issue of getting stuck in local minima. As much as we have more information in the current electron density, we have more information minima also in the current, and you can run into the issue of getting stuck in local information minima. And in a way, with very high resolution densities, if you manually build, it's fine. You can see the density, and by hand, you can start plugging in these helices into these densities. But if you want to do something computationally that is automated, then essentially you run into the issue of this structure and this structure are both being fitted to the density, almost with comparable cross correlation, almost with very similar cross correlation. But this is correct and this is incorrect. This is because there's a local minima issue that has come into these simulations. And then that was a point of major problem with molecular dynamics as a whole entity, where Monte Carlo oriented tools started becoming a lot more popular. Something like Rosetta. So David Baker around 2013 already started writing papers highlighting these kind of issues with molecular dynamics and molecular dynamics flexible fitting. So then we had to get around this. And so we came up already, this was around 2016, towards the end of my postdoc. We started doing simulated annealing in the density space. This was to kind of start, making the refinements better. So in a way, we started out with a filtered version of the density, which had a lot less information, and then gradually improved the density. It's called low pass filtering, refinement with low pass filtering, and it became quite successful. Thereafter, this kind of work has been reproduced across a number of by a number of scientists in a number of tools. In fact, if anyone uses Isolde today, which is a way of manually refining structure, flexible fitting today happens with this principle. So people learned from us, and then they have implemented that in a number of different molecular dynamics simulation tools. So so then we overcame that issue, and now we have started getting also very good sidechain alignments and very good sidechain refinements with that. So then after that, we also found out one more thing. So we were talking about local resolution, the fact that, the cryo-EM map has heterogeneous density around. We could correlate the fluctuations, the real structural fluctuations of an ensemble of structures with the local resolution of a density map. So this became very, very transparent. So one of the reasons, a conventional structural biologist often uses what you call a single density, single data paradigm, where if you have a single piece of data, you often try to interpret that with a single structure. But the reality is that there is an ensemble of structure, there are multiple structures, the average of which is being seen by the experiment. And in principle, the reason for utilizing something like a molecular dynamics or a Monte Carlo code is to get that ensemble, which nowadays is getting more and more appreciated. 5 years back, it was not as much. In fact, when the high resolution cryo-EM data came up, a lot of people started telling we don't even need molecular dynamics because we are seeing the structures very clearly. Having now, of course, the same people understand that the interpretation of that cryo-EM density does not lie in just one structure, rather an ensemble. And molecular dynamics is a very, very good way of seeing those ensembles. So, yeah. No. Any questions so far? Like everyone is so silent. I feel scared when people are silent. So, any thoughts? I think anything the online colleagues are thinking about they can share. They can put it on the. I don't know if they can. I can't see the
Oh,
chat.
I can see if it's anybody.
Yeah. So then very quickly we started utilizing this in the EMDB challenges, and so these are a number of scores that people in the electron microscopy community they like their model to have. Normally, the greener they are, the better. So we appeared in a number of these challenges, and red. These are the scores that we had. They all more or less looked very respectable on the higher end. So we were in multiple times these kind of methodologies used by us or others have put us in the top ranks of some of these communities. So then, of course, and this tool is available. This is the website. Anyone can download it and start using it. It's very, very straightforward to do it. Of course, you need to know molecular dynamics simulation a bit. You need to have a little bit of a computational chemistry background. Otherwise, it's difficult.
Is it a standalone code or is it interface?
It's all inside NAMD, so you can just utilize that for NAMD. So now the next thing, okay, there's can we do better? So let me not get into the maths of this. Let me actually show the movie first, and you will realize. Okay, so now what happened is if we have this, if we have a molecular dynamics flexible fitting simulation, a lot of the times what happens is we use our chemical intuition to fit structures into densities. But what happens if we don't use chemical intuition, if we don't use chemical restraints, if we don't use the idea that there is no chemistry present at all, then this is what happens. You get a really, really contorted structure. Because what you have done is, you have removed all your chemistry information, all the molecular dynamics force field is not a part of this information. You're only using information from the experiment, and that's it. And then trying to optimize based on that. Then you get really, really bad structures. What you end up getting is, so what are we doing? We're telling there's a bunch of atoms and put them inside densities. And what is happening is, these simulations are optimizing or maximizing, trying to put as many atoms inside density features as possible without even thinking about connectivity. So this problem remained in molecular dynamics flexible fitting because of a particular issue, is that the forces that are present in the simulations had to be changed in a way. So then again getting into, and these problems are lesser. For this is on the x-axis, I'm showing the percentage of incorrectness in our model. This is what you call overfitting. For larger systems, it's actually smaller. For bigger systems, it's lesser. In fact, not only that, for different side chains, for these three side chains, if you do flexible fitting, these problems remain the most as opposed to other side chains. In fact, any current flexible fitting technique has this, not just our tool. The way this is done in a more traditional fitting structure into the data sense, that issue remains. So yeah, these three are problem residues. Then we realized that, and then we kind of overcame this issue. Again, let me not show the maths possibly. So just look at this equation. So, and compare this to the original equation of how we were forcing atoms into densities. In this particular case, what did we do? We took our density and we simply did a linear, we put the density on a grid and we did a linear derivative of that density. What that would do is, if I have two different atoms, if my density features look like this, and if I have two different atoms, this density will put both the atoms in the same low energy to reach an energy minimum by definition. As opposed to that, now we have come up with a different kind of an energy bias where it looks like a spring, essentially. It's a harmonic energy bias. So if I have two atoms, One will go to a lower energy slide and another will be, and this will actually improve the correlation between my atoms and the underlying density features. Here, this would put both the atoms within the lower density feature. That's what I was showing you in the previous thing. So with doing that, we kind of came up with another way called multimap of fittings. Now we're actually getting real ensembles. And one of the good quality about ensembles is that you can start computing energy changes or free energy changes. If you have populations, then the negative log of that population is actually energy. Clearly from cryo-EM information, you can say what are the energy differences between multiple states and how do you extract that, that information. Now we can do that. Back in the day, we were not being able to do it. So now we can actually not just fit a structure into a density, but during the process of the movement, we can tell you the energy cost of that movement. Any traditional flexible fitting will not be able to do that. And this energy cost is important because biology understands energy. Biology does not understand a stationary structure. Once you move from one structure to another, how much energy is required is what is important for the cell, so to speak. So now we can do that. So from cryo-EM data to structure to ensembles gives you energy. Otherwise, you will not be able to have that. So that's kind of a strength of these kind of methodologies. Then, of course, again, we have and this also, now we also get around this issue of, because we're using a new kind of potential, the issue of overfitting structure to the density goes away. And we actually start getting better, what you call mole probability scores or better stoichiometry codes. Again, this code is also available and people can use that. So then finally, we did one more thing. Folding, unfolding is a kind of issue that we often face when molecular dynamics, when you start to kind of fit structures into density. Disordered structures, for example, is really of importance. Disordered parts, intrinsically disordered proteins. That is so. And traditional molecular dynamics, folding, unfolding transitions are very difficult to gauge because the amount of energy that is required to go from a folded state to an unfolded state, the energy barriers are really high. You will not be able to sample that. Monte Carlo is possibly the only way of doing it. That's why tools like Rosetta does it really well. So then, but we wanted to recreate that in the molecular dynamics setup. So we came up with a methodology, Ken Dill, one of the fathers of protein folding. So together, we worked together to came up with a methodology called a Bayesian inferencing methodology, in fact, called MELD, where without getting into too much of the details of what Bayesian inferencing means, we use experimental data as a prior. In all the examples that I showed you so far, experimental data was used like a spring, so to speak, and you were dragging your structures towards that. Now your experimental data is actually like a prior. It affects the probability of your system, not necessarily the energy of your system. So if you bias the probability, by definition, you will bias the energy. That's the thought. So now we started, and this so we use this experimental data as a prior. And in doing that, the implementation of that is you use a number of springs here. This E restraint becomes the same definition of spring that I showed you before. But we are not, our bias is that of a probability. We are not doing derivative of energy with respect to position, rather derivative of probability with respect to position, as a result of which you get a force, which is a much softer force. So that's what Bayesian inferencing, in fact, tells us in a way. So once you do Bayesian inferencing, and so now you introduce the same potential, but not directly, rather using a Bayesian inferencing framework. And that this let me not get into the weeds of this. Now you can do something like this. You can take electron tomography data. So what I'm showing you here is, this is an NL63 spike protein, where simultaneously being fitted to experimental data from two different sources, because in principle I can do that now. There's a very high resolution cryo-EM map that is not there in the density, to which the crown of the spike is being fitted. This is a low resolution electron tomography reconstruction, to which the entire system is being fitted. And both these experiments are simultaneously, the data is simultaneously being fed into the simulations, not just one. You can actually do multimodal fitting with that. which a single molecular dynamics will not be able to do. You can actually use cross-linking for this, NMR for this, any kind of information. Each one of them becomes this E restraint in the, just to go back. This E restraint need not come from I goes from 1 to 100. This can be 100 restraints from very different kind of experiments. It doesn't have to be the same experiment. And that is where we actually can start, mixing things together in a way. And yeah, so here we did electron tomography. This paper just got accepted this morning, so I'm very happy about it. In fact, this is WAS data, WACHU's cryo data. So okay. So now then, why would you? And again, this tool is also available if anyone wants to use it. Why is this so important? Because I was telling you about ensembles. So what is one of the first structure for which, in fact, they got the latest Nobel Prize? David Julian got it, not Yifan Cheng, but was TRIPV1. So TRIPV1's cryo-EM structures were some of the earliest high-resolution structures of cryo-EM. In fact, this was the structure that was seen in one day in one cryo-EM map, and this was the structure that was seen in another cryo-EM map. What we see is a distribution of all of these structures, all of which interprets the overall electrophysiology of a certain protein. Cryo-EM picks up either one of them or two of them, but and in fact you have to do separate experiments to get these, which is correct. You need that because that way you don't know if your focus classification is biologically relevant or not. But starting from a data that actually would give you this structure, which is the most probable structure representing that data. If you get an ensemble, a Bayesian, in a Bayesian sense, you can actually see rare portions of that ensemble, which you might pick up in another piece of experimental data. So you can get a number of representations. Some of this might not be very, very good, so you don't worry about it. You have to do orthogonal experiments to treat it. But the ensemble representation gives you this. It gives you an interpretation. You don't overfit your intuition to it. You get what is most probable, but something that is low probable structure might also mean something biologically. In this particular case, it means that there is a transient beta sheet. This beta sheet need not be absolutely stable. If I just show you this structure, you will feel like there is no dynamics here. This beta sheet is this beta sheet. But turns out that this beta sheet can undergo an unfolding, which will make compromise the local resolution, but you would never have, as a structural biologist, a way of telling that, whether this is a beta sheet or not. But in these kind of simulations, you get alternate interpretations. You don't have to imagine anything. The simulation shows you that. Okay, so now then, and then again, the other thing is at a cryo-electron tomography level, once you do subtomogram averaging, you get multiple different conformations of the same spike. And as you can see, our simulations are fitting to the spike. Yes, for most of the times, but they're also going to other conformations. That is not necessarily seen by the experiment. So it's not just fitting to it, it's inferring to it. So if there are WA got us 7 different electron tomography maps, and we had to figure out an ensemble of dynamics. Each one of them is just 7 structures, but the entire NL63 virus capsid or protein has a huge range of dynamics, which actually has a population. And this population, like you do focused classification and do subtomography, basically reconstruction for each one of those, we can actually do focused classification of the molecular dynamics simulation exactly in the same way as done in cryo and come up with a very strong list. In fact, this is the first paper that just came out today, which will show that a distribution coming from cryo-electron microscopy will actually overlay with the distribution coming from molecular dynamics simulation. We were waiting for that system. Spike has that system. And not only that, certain part of the distributions are actually more probable, which is around 40 degrees, which cryo-EM showed and MD showed, molecular dynamics showed independent of cryo-EM. And that is where a number of questions are now coming up. Can we see molecular dynamics kind of heterogeneity in cryo-electron microscopy and vice versa, which was the discussion that I was having with James before the meeting. So okay, good. So now I will not bore you. We can literally now do things that are seen in electron tomography, let go of electron microscopy. And these are huge simulations. So in order to simulate an entire vesicle, you are actually looking at 100 million atoms, but we can do that. We can do those kind of simulations right now. So okay, good. And again, these were where the maps were. So it's let's see if we have anything. It's good. So I think let me now stop this facet. I had some more development that had to do with QMMM because I wanted to show the quantum aspects. There are certain parts of the, one of the biggest problems in cryo-electron microscopy is the local resolution of the cryptic binding pockets is so low that you cannot attribute drug binding to certain pockets without prior knowledge. Unlike crystallography, where things were known very well and one could run very long crystal screens, any biopharma would do that. Cryo-electron microscopy is not there. In fact, the latest electron microscopy challenge was protein small molecule interactions. How would you address that? So then the point is there, what we did is we have a way of doing certain part with quantum mechanical accuracy and other part with molecular mechanical accuracy, all of which is working under the influence of experimental data. So that is essentially not just. So now I break the assumption that I made in the first place. In my first slide, I said that atoms are balls and bonds are springs. I say, okay, for certain parts, we can get around that assumption. We can actually do real quantum chemistry, surround that with molecular dynamics, and do it all in the presence of experimental data. That is where I think the working with NW Chem would open new areas for us in terms of figuring out how does drugs look in binding pockets. I think I had, okay, we have some GUIs to use it and all that. See, in fact, yeah, just to kind of allure you, we also have very easy, straightforward input, making, this is for games and for Gaussian as part of NAMD. We can have a /NW Chem here if we work together. So now then, again, so this is like a density, and somewhere here, this drug should, this drug should get into the density, and gradually we can track what are the residues. So it takes some 1 nanosecond of QM-MMMD simulation. So because this drug is actually treated at a QM level to be able to fit it in the density, and we also get ensembles in our cryo-EM challenge. With the drugs, those are separate slides. I can show what are the multiple poses of the drugs that can be seen. And in doing it's very difficult to have an unambiguous assignment of drugs in this. Now we can potentially do that. And together with QMMM, we'll be able to do it much better. So if we can have an NW Chem interface here, then possibly we can work together. If there is any protein ligand problem happening here, we can definitely look into that together. Okay, we also did things with X-ray crystallography, but let's not. But essentially, you re-refine. You don't just refine once. You keep refining the structure and the density iteratively, because in X-ray crystallography, the phase problem is not solved. In cryo-EM, we know our density. The density is given to us. In X-ray crystallography, we simply have 2D patterns of diffraction. So we have to keep refining the density and the structure hand in hand till we get the final structure density fit. It's a much older field. We can do that. Of course, there are certain, so not only at a local side chain level, also at a complete, all atom level. So you can take the same thing and push it into X-ray crystallography. So, okay, now then, and this is a very, very highly timely area. We are not the only people who does that. There are a number of molecular dynamics based, of course, non-molecular dynamics based teams have been there all around. There is a number of molecular dynamics first principle physics people who are doing this. Some of them are also ensemble methods. Not all of them are ensemble methods. So you might or might not have looked into it. But yeah, there's Rosetta EM Fold and MD. People have done a lot of things here. But it's an area of absolute interest in integrative modeling. So, okay. With that, then let's move on to some applications of these. I would skip, how much time do I have? I have only 10 minutes or even lesser than that, right? In the interest of...
Just a little bit more.
So in the interest of time, let me just show you some of the best applications in my lab that has happened. Let's not. So we actually found the structure of AstraZeneca's COVID vaccine when they kind of were having trouble in figuring out why was this vaccine clotting blood. So we determined using some of these methodologies how to we reconstructed the Oxford AstraZeneca vaccine and then started doing. Okay, this might be one thing. Like often when you do reconstructions and do models, you will see that you end up getting very bad reconstructions with bonds moving through, getting through each other, and the chemistry, like your model is really, really bad. Sometimes you'll also find in PDBs, in submitted PDB files, these kind of artifacts. So how do you get around that? We came up with a way of using free energy perturbation to start growing atoms using Monte Carlo based techniques so that they don't kind of bump into each other. It's called a long bond eliminator, and this is George did, and so you will not be able to have these kind of problems on the left. A little bit of technical thing that we found while we were doing these large multimodic systems. Okay, so this is again quickly. Sorry, I just wanted to highlight the link. There's a link to that also. But all of these are for molecular users of molecular dynamics at one level, because our code is a molecular dynamics tool. Okay, then once we did our calculation, actually this PDB is now released. It's an old slide. We could see the electrostatic potential on the outer surface of this ChAdOx, of this particular vaccine. And we found that the AstraZeneca vaccine is actually more electronegative than the Johnson & Johnson vaccine. As a result of which, it ends up binding a particular cytokine called PF4, and this is the binding mechanism. The movie runs. Okay, accelerate the movie. So the lower part is a cryo-EM structure that we determined. And on top of that, these cytokines are binding. Why is this cytokine binding will be my last slide, and then we'll stop for the day. So we found out that there's a very weird electrostatic interaction between this cytokine protein and the virus capsid and the vector. And then we came up with a reason. Again, this paper was accepted in 2021. Now it has been cited like close to 200 times now. So what happens is that why was this vaccine clotting blood? If that question was asked to us, so normally cytokine PF4 is a kind of cytokine. Cytokines are not supposed to get into a lymph node where your platelets to trigger platelets, which causes blood clotting. PF4 by itself will not be able to reach the lymph node. However, the vector of the vaccine is designed to reach the lymph node. Now, if there is an electrostatic interaction between the vector and the cytokine, which we also established using SPR measurements, then the hypothesis is that this cytokine basically hitches on the surface of the vector and then goes to a place in the cell where it was not supposed to go to. And once it goes to that place of the cell, then it triggers antibodies that are anti-PF4 antibodies and essentially triggers an autoimmune response from our body, which causes the blood clotting to happen. Then, of course, why is it not happening to everyone? It's like 11 out of a million. That was the statistic of blood clotting. 1 out of a million being the statistic of you being hit by lightning. So 4 out of a million was actually not. It's 4 out of a million. 11 out of a million is your probability of being dead in a car crash or something like that. So it's even lesser than that. But then why is it not happening to everyone? Because then we found that anti-PF4 antibodies are very, very rare. So it's very possible that this vaccine is distributed in 194 countries, and it's very, very possible that the PF4 is going to this weird parts of the cell for almost everyone, but not everyone carries these kind of antibodies to create this kind of platelet responses, and that's why it's not happening everywhere. So then, potential way of using this vaccine could have been that we propose is screen people for antibodies before you give them the vaccine. If you don't have this anti-PF4 antibody, then give it the vaccine, then potentially this mechanism of blood clotting is not working. But that's too expensive to do at an industrial level. So that could not be done. But then, of course, we worked with AstraZeneca to redesign the surface of this vaccine here. And now, starting from the original vaccine surface, we did computational modification to actually bring down, to come up with mutants where the binding spots are a lot less. I cannot disclose the mutants. Around the beginning of 2022, we handed over 31 computational designs to them. And that was our deal, that I will not know which one will they pick after that in their subsequent distributions. And then, yeah, so that was a great story about how academic research and industry works hand in hand, literally over a period of 6 months to redesign a vaccine in the market and change it to move things forward. So yeah, so that's kind of the, let me kind of stop the talk there. I had some other slides, but I think I kind of overestimated my speaking capabilities. But it's okay. The basic take home is that there are a number of tools using molecular dynamic simulations that are available today that gives you an ensemble representation of the experimental data, not just one experimental data, but experimental data of different sparsities that can be combined, brought together. Number two, and then number three is that they have like very high-end practical applications. And in a structural biology unit such as here, where you are actually exposed to data of different modalities, which is very similar to my background, the center that I belong to, we have almost everything barring the amazing mass spec that you have here. So to kind of, come up with methods, to come up with algorithms that can bridge this, have an integrated view of structural biology rather than a single experiment or a single model view of structural biology. I think that potentially would be the biggest take-home message. With that, I'd like to thank you for your time. Thanks.